Prokaryotes can alter DNA sequences through transformation, transduction, conjugation, and transposition. Viruses often recombine genetic information, and all of those changes are subject to natural selection. But ultimately, it's the processes that increase genetic variation that are conserved. So when we talk about bacterial variation, the first thing we'll look at is conjugation. It's the closest thing we have to sex with bacteria. And again, when it comes to sexual reproduction, that is a eukaryotic form of reproduction. But when we look at this, we can have two individual bacteria here, and the one can send a pilus to the other, and it's through this pilus that DNA can be transferred to other bacteria. And a lot of times it's this plasmid that can be sent over. So then ultimately, one bacteria can give another bacteria genetic information. Another form of bacterial variation comes about through transformation. And again, with transformation, we have this naked DNA or a plasmid in the environment. That can get there in various ways. Sometimes bacteria can shed plasmids. Other times when they die, plasmids are available in the environment. And a bacteria can pick that up. Sometimes we'll look at that through heat shocking when it comes to genetic manipulation. But ultimately, bacteria can pick up loose DNA from the environment through transformation. Another form that will lead to bacterial variation in their genome is transduction. So we have this virus, a bacteriophage, infecting a bacteria. So this phage will secrete its DNA into its host. And in that process, the bacterial DNA, the bacterial chromosome, will get lysed and chopped up into lots of bits, and it will kind of intermingle with the viral DNA. In that process, we have these bacteriophages being reassembled, and some of the bacteriophages may have collected some of the original bacteria DNA. These new bacteriophages, these viral particles, contain some bacterial DNA that can then get injected into new bacteria with different chromosomes. So in that process, you don't always lyse up the entire chromosome. In this case, some of the original bacteria DNA gets put into a new bacteria, and it absorbs that into its chromosome, and now it has added variation. Another form of variation comes about through transposition. So we have our bacterial chromosome, and it has various genes. Now you can have these transposable elements, and these are like jumping genes. So they can be copied and leave the original chromosome, and then they can be put into plasmids, and then they can be transferred through a lot of the different processes we had just talked about with conjugation and transformation. So that plasmid with this bacteria from the original chromosome can get transferred into the environment or to other bacteria. Now, this transposition in transposable elements is not exclusive to bacteria. When we look at our own genome, 45%, again, only 1 to 2% of our genome is coding for proteins, but 45% of our genome is made up of these transposable elements. So what's really interesting here is that some of these transposons are retrotransposons as well, similar to retroviruses, where the original DNA does get transcribed into messenger RNA or into RNA, and then that goes through reverse transcriptase through this enzyme, and it turns it back into DNA, and then it can be inserted into other areas of the genome. And where it gets inserted is what part of the DNA it comes from, and where it gets inserted has a huge effect on the overall genotype and phenotype of that organism. So one thing that's really interesting about transposition in humans is that there's a lot of cancer research, going on where there's a transposition of the promoter sequence, and that promoter sequence getting pulled out could get put into in front of proto-oncogenes. And again, these are the gas pedals for the checkpoints in cell division. So if you take a promoter transposable element and put it in front of an oncogene, all of a sudden you're just putting the gas pedal down, and that can cause cancer. So when we talk about viral variation, obviously we have mutations. So when we think about coronaviruses or COVID-19, a lot of the new strains come about because there's an infection. There aren't all those checkpoints to make sure everything is copied correctly. So in the process of that replication, mutations can accumulate, and now we have a new strain of that virus. We also have recombination, and this is a little bit different than our crossing over recombination with eukaryotes, but we're recombining the strains into a new hybrid strain. So if similar strains of viruses infect a host at the same time, in the recreation of the virus, some of these individual pieces of DNA can be reassembled into a completely new strain of virus. We can also have recombination where we have these segmented RNA viruses that also are infecting, and it's these reassortment of all these segmented RNA segments, this genetic information that can get reassorted and reassembled into new variations of the viruses. Again, this is what kind of makes the flu virus so hard to stop, because influenza goes through this process of reassortment. We also have retroviruses like HIV. When it goes, the RNA gets infected here into the host cell, it goes through reverse transcription and gets buried into the DNA. But then in this recombination, we can have all new varieties. And again, this can happen over and over and over, where we recombine all of these different versions and we continue to evolve the genome of these viruses. So moving on from that, one thing to keep in mind is that diversity equals stability. That comes into ecosystems, but it also plays a major role when we start thinking about all life forms. And this gets into this idea of antibiotic resistance. So let's say we have this bacterial strain and it has mutated. And we try to kill this bacterial strain through antibiotics. So we give it a dose of antibiotic and it kills some of the bacteria. We keep on going with our treatment and it kills more. We keep on going with our treatment and it kills more. However, these mutated strains may be resistant to have a mutation that makes them resistant to that antibiotic. So even if we keep on treating it, these guys that are resistant can multiply, and now we have a very much more serious version of this bacteria strain because we cannot kill it. So then we try a different strain of antibiotic and it doesn't work. We try a different strain, 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 it doesn't work. And now we're in very serious trouble because we're basically out of medicine. And we're back to where antibiotics weren't invented before, and it's up to your immune system to get rid of these particular resistant strains. And again, sometimes that resistance comes about in different ways, and it could be through plasmids, which can be transferred from one to another. Again, a lot of that comes back to the whole evolutionary idea of survival of the fittest. If the environment changes and wipes something out, those that are able to survive are going to also reproduce. So this idea, again, diversity equals stability. If we have a variety of organisms in a place with a variety of genetic, whether it's one gene pool or whether it's an entire ecosystem, diversity equals stability. So we keep on adding diversity We keep on adding diversity, we keep on adding diversity. This gene pool is really complex, or the ecosystem is really complex. So when a disturbance comes along, it barely affects the overall gene pool or the overall environment. So again, when we think about this process, it is these processes that are transforming and adding diversity to the gene pool and to the ecosystem. So when we think about the gene pool, again, we might have a common ancestor, and it's various processes that are transforming the genetic code over time, and that leads us to diversity. And again, it's not necessarily the organisms that matter, it's really the processes that matter, that add to the stability of life over time. So when we think about overall diversity, how does that come about? In eukaryotes, we have crossing over, we have independent assortment, we have random fertilization. All of those processes happen through meiosis and then sexual reproduction. When we talk about bacteria, we're talking about conjugation, transformation, and transduction. Transposition happens with bacteria and eukaryotes. Recombination, although we often use that term when we're talking about crossing over, recombination is also a viral concept. And with all viruses, bacteria, eukaryotes, all of them are subject to mutations, and it is these processes that make life stable over time because they add to diversity. So again, prokaryotes go through transformation, transduction, conjugation, and transposition. Viruses can recombine genetic material. All those changes are subject to natural selection, but it's the processes that increase genetic variation that are conserved over time.